Good morning, friend. Welcome again to the e-learning class of biology. We'll continue with the topic control coordination. First part we are dealing with nervous control coordination. What we studied up to now, quick revision we'll take. We studied nervous system of Hydra and nervous system of Planaria this year. Last year we already studied nervous system of cockroach. We took the comparative account of these nervous system. Hydra we have a nerve net. Planaria will have nerve ring and a nerve net. Similar we'll have in case of cockroach, nerve ring and a nerve net type of a nervous system. Even we discuss neural or nervous tissue present in animal, and even we discuss how the transmission of the impulse takes place within one neuron and from one neuron to the another neuron. Places are called as synapse. And we have two types of synapse: mechanical, electrical synapse, and the chemical synapse. Chemical synapse are with the help of neurotransmitter. And now we want to discuss next part of a chapter, that is actually revision what you studied in earlier standard, but we are going to little detail here about nervous system of a human nervous system. We want to discuss in detail. Here we are going to discuss that human nervous system is well developed and complex in nature. When we say well developed and complex, well developed and complex, we compare it with the help of other animal. As compared to the other animal, the nervous system here is made up of different organs, as well as will have a complexity in these organs. That way we say it is well developed and complex in nature. For our study purpose, it is broadly classified into the three parts. CNS called central nervous system, PNS called peripheral nervous system, and ANS called autonomous or automatic nervous system. What is CNS? In short, we'll discuss and we'll proceed. A diagram that I'm giving here. This is the central nervous system. Nervous system present in this central axis is called a central nervous system. We'll have a two main organ here to deal with in central nervous system: brain and the spinal cord. Then we have whatever blue color are shown here. These are the nerves which start from either spinal cord or brain, and they go to the different parts of the body. Nerves we already discussed. It is the bundle of nerve fiber, and these nerves constitute peripheral nervous system, and some of the organ which automatically works in the body, like heart and kidneys, etc. Nerves are supplied to this particular part. They do the work automatically, and this is called autonomous or autonomic nervous system. One by one, we want to discuss. So first, we'll discuss central nervous system. As I already told you, this is the central nervous system made up of this brain and in the vertebral column. These are vertebrae actually from outside. In the vertebral column, we'll have these spinal cord. These two will form central nervous system. Simple definition I'm giving. Nervous system present along the central axis or mid dorsal axis of the body. See, this is present from dorsal side as it is characteristic of vertebrata. And the one which is present from dorsal axis of the body, the one which is for central nervous system, in short, we call CNS, central nervous system. As I already told you, it is made up of two organ. Number one is brain, number two is spinal cord. This is brain, this is spinal cord, I told you. And the one which derived from embryonic ectoderm, we already discussed. Every time we'll discuss the embryonic origin. These develop from embryonic ectoderm. Both are covered by protective coverings in addition to the bony covering. No doubt we have a bony covering that we discussed in 11th standard. Brain protected by cranium, spinal cord protected by these vertebral column, vertebrae. And in addition to that, both brain as well as spinal cord, they are protected by protective coverings called as meninges. This lecture we are going to study meninges and cerebrospinal fluid which is present in central nervous system in detail. One by one again, first one we'll discuss meninges. Diagram that I am giving. See, this is the brain from inside. This is the total head with the skull, and here the brain inside. This much section I have enlarged here for your understanding. Diagram is not expected, still I'm showing the diagram. See, if from outside we'll have bony covering. This is the bone, and then we'll have three coverings from outside to inside. These external protective connective tissue coverings around central nervous system. Remember, not only brain, but brain and spinal cord. This external protective connective tissue covering around CNS is called a meninges. Remember, singularly called a meninx and plurally called a meninges. Simple definition. We'll have a short note on this one. So, external protective covering, which are actually connective tissue covering around the central nervous system, and these are called meninges. Three are there, remember. This is the way to remember. Outermost, middle one, and innermost one inside will have a brain. Even we'll have a gaps in between these. This we are going to discuss. Outermost protective covering, this one I'm talking about, is called a dura mater. Spelling mistake is here. It should be M-A-T-E-R. Actually, we pronounce matter, M-A-T-H-E-R, but it is matter, remember. So the first one is called dura mater, outermost protective covering, the one which is tough and non-vascular in nature. Vascular, when we say, we'll talk in terms of either supplied with the blood vessel or not. This is the outermost which is not supplied by blood vessel, the one which is tough, protective, strong, and the one which is thick in nature. So as compared to these remaining two, I'll repeat, outermost is called dura mater, this one. Middle one is called arachnoid matter, this one. And the innermost one is called pia matter, this one. Remember, out of these three, outermost is thick and the one which is fibrous. Can you guess which fiber it must be? Yes. The one which gives the strength is a collagen fiber. This tissue is made up of more amount of collagen fiber, remember. The one which is attached to the inner side of the cranium. See, this is the inner side of a cranium. This is attached to the inner side of cranium. And the one which is separated from next layer, arachnoid method, with the help of a space called subdural space. Remember, subdural, when we say below the dura mater, this is called subdural space. The one which is filled with the serous fluid we'll discuss. But remember, the outermost covering is thick and non-vascular, and the one which is fibrous. And because of a collagen fiber, this will get additional toughness. Definitely, it will be protective. All three are protective, but additional duramatter will be protective in nature. Additional work will be there for some other. Second one we'll discuss. See, this is from here to up to here, and this is called as middle one, the one which is a thin again, the one which is non-vascular, little bit, but connective tissue with wave-like appearance. Remember, see, this is wave-like appearance. From here to up to here, this is the space we are talking about, in correct level. This is the arachnoid matter, and the one which is with wave-like nature, separated from next layer, quadripia matter, with the subarachnoid space. See, this is dura matter, subdural space. Arachnoid matter, subarachnoid space, remember. Okay. So this is something about wavy nature of second one, and this is called arachnoid method, remember. Okay. Third and innermost one is called as a pia method, the one which is innermost, the one which is very delicate. See, the one which is thin and the one which is delicate, but the one which is highly vascular in nature. Vascular means supplied with the blood vessel. So whatever the nourishment is to be given to the remaining layer, even to the brain, that is with the help of blood vessel present in pia method. Innermost, the one which is delicate, the one which is highly vascular membrane, and the one which is in close contact with CNS. So like what we have, dura method with subdural, arachnoid method with subarachnoid, we don't have a space between brain and pia method, remember. So two spaces that we have below the dura method, below the arachnoid method, maybe in spinal cord, we'll have above the dura method space, this is called epidural space that I'm not mentioning here. So remember, simply we'll have most probably multiple choice question. Three, the one which are from outside to inside, or they may be asked from inside to outside. Outermost called dura mater, middle one called arachnoid mater, and inner one is called pia mater. Okay. And then we'll have a fluid in these particular spaces present in the matter meninges. In addition to the whatever gaps are present in the brain, And these gaps are also called ventricles, remember, ventricles. And the fluid present here, this is called as CSF, we call cerebrospinal fluid. Name itself is the meaning, the one which is present in brain as well as around the spinal cord. And this fluid is called as cerebrospinal fluid. Remember, the one which is lymph-like extracellular fluid. Lymph like blood lymph we are talking about. The one which is the fluid present outside the cell, so extracellular fluid. And this fluid, here I'm showing actually the diagram of a brain with the gaps here in between. See, these are the gaps present in the brain. These are called the ventricles. Here we have a third ventricle. Here we'll have lateral ventricle. Here membrane again I'm showing here in detail. And whatever the gaps are here, these gaps, blue color which is shown here, even around the spinal cord and inside the spinal cord again from this canal because central canal we'll have this fluid, and the one which is called cerebrospinal fluid, the one which is secreted by two parts, remember. One is a part of a brain called choroid plexus. I'm going to remind you when we'll study brain. This is the part of a pyometer actually, choroid plexus, the one in addition to the type of a cell lining this spinal cord, these are called ependyma cell. And this one will secrete this particular fluid because cerebrospinal fluid. I'll repeat two parts, choroid plexus and ependyma cell. Choroid plexus is a part of brain we'll discuss. And ependyma cells are present inner lining to the spinal cord. They will secrete this particular fluid. Okay. Where it is present, as I already told you, inner to the subarachnoid space, ventricles of a brain. What is ventricle? I told you gaps present inside the brain are called as ventricles of a brain. Don't confuse with the ventricles of a heart, remember. Here, gaps present inside the brain we are going to study separately, and these gaps are called as ventricles of a brain. And even spinal cord will have a canal, I told you, cavity, I told you, and this cavity is called as central canal of a spinal cord. All these are filled with the fluid, and this is called cerebrospinal fluid. The one which will come out of a three openings present in the roof of a medulla oblongata. See, here we are going to discuss the part of a brain. This is called medulla oblongata, and the one which will come out of this particular brain part to the lower part with these three openings present in medulla oblongata we'll name afterwards, and the one which will help to drain cerebrospinal fluid from brain to the outside, remember. So three openings are there that will drain from roof of medulla oblongata to the rest of the part. This is something about introduction. Now, 3 4 point we'll discuss. Next point is the nature of this fluid. I told you this is slightly alkaline fluid with a little more specific gravity than water. Specific gravity is 1.005. We'll have multiple question. An approximate quantity is 100 to 120 cubic centimeter of cerebrospinal fluid, CC, and it is present in and around central nervous system, we say. Okay. The function wise, the one which will act as a shock absorber. So whatever the shock that we bear for the head, never the brain get damaged, remember, because the shock will be taken by meninges, no doubt. In addition to the meninges, this fluid will help for absorbing the shock. That way it is called a shock absorber. And that way I told you it protects the brain from any mechanical injuries, remember. So never, if you stuck to the head, brain get damaged because the shock will be absorbed by cerebrospinal fluid. Next, it maintain the constant pressure inside the cranium. So whatever the pressure that is required inside the cranium, because brain again is very soft, remember. So to maintain that brain structure, to avoid the drying of a brain, and that pressure from outside as well as inside will be maintained by cerebrospinal fluid. It will keep the brain moist. As we already said, the one which is fluid in nature, the one which contain water on a large amount, the one which will keep on supplying the nourishment to the brain, and that way it will keep the brain moist in nature. It will avoid drying of a brain. This is called desiccation of a brain. So this never happens. We'll have a abnormality where cerebrospinal fluid creates the problem. Okay. Next one, it helps in exchange of nutrients and waste. As I already told you, pia matter, we have a vascular nature. The nourishment will be there up to the pia matter. Now that will be carried with the help of this fluid to the different parts of body, that parts of a brain. That way it will help for exchange of nutrients. Even nutrients in terms of food will be supplied, waste will be removed from the brain, and the work will be carried between the blood and the brain with the help of cerebrospinal fluid. Remember, okay. Next one, it will help in supplying of oxygen to the brain. This is main and important function. We all know a brain require much amount of oxygen. And if at all brain do not get oxygen for more than 5 seconds, we suffer with the problem, remember. So the one which is supplied constantly from the blood to the brain, exchange of a material will be carried through this cerebrospinal fluid. That's it. This is something about cerebrospinal fluid. We'll summarize what we studied today. We'll stop because part of the brain we want to discuss, that we'll discuss in the next lecture. What we studied today, we started with nervous system, human nervous system to discuss with. And in the human nervous system, we discuss that we have a three part: central nervous system, peripheral nervous system, and autonomous nervous system. I told you before we go into the details of a nervous system, two important structure we discuss. Number one, it is meninges, protective coverings around the brain. Three are there, I told you. Spelling is actually MATR, matter, but we call mather, remember, MATHER, mather. You can remember the names from outside to inside or inside to outside. Question will be specific, remember, in a CET and a NEET examination, because generally we remember from outside to inside, but question will be from inside to outside. So which all three protective layer from inside to outside? Pia method, arachnoid method, and dura method, remember. In addition to that, brain will have a protection of a cranium, and the spinal cord will have protection of vertebral column, remember. Three meninges we said, we'll have two spaces. Below the dura mater will have subdural space. Below the arachnoid matter we have arachnoid, subarachnoid space. And I told you in case of a spinal cord, not in brain, above the dura mater we'll have a space, and that is called epidural space, remember. Okay. And all these spaces, in addition to the gaps present in the brain, these are called ventricles of the brain. And they all are filled with the fluid. And this is called cerebrospinal fluid, CSF, CFL, CSF. And this fluid is a lymph-like fluid, alkaline in nature, with a specific gravity, 1.005. And the one which is produced by choroid plexus and ependyma cell, the one which will play important role for absorbing the shock, avoid dry desiccation of the brain, exchange of material in form of supplying nutrient and removing the waste, and main and important, I told you, the one which will act as shock absorber. That's it. This is something about today's lecture. Today, two point will be, two short note will be there. Right short note on meninges and right short note on cerebrospinal fluid. Or question will be specifically asked on functions of cerebrospinal fluid. That's it. Here we are up to. Next lecture, we'll discuss with the central nervous system, and we'll start discussion with human brain to discuss in detail. As we already said, human brain is highly developed in nature. How this developed nature is there, that we'll study with the help of next lecture. Okay, we'll stop. Thank you very much.